6. Nucleic acids and protein synthesis. Nucleic acids have roles in the storage and retrieval of genetic information and in the use of this information to synthesize polypeptides. DNA is the molecule of heredity and is an extremely stable molecule that cells replicate with great accuracy. The genetic code explains how the sequence of nucleotides in DNA and messenger RNA, mRNA, determines the sequence of amino acids that make up a polypeptide. In eukaryotes, this involves the processes of transcription in the nucleus to produce mRNA, followed by translation in the cytoplasm to produce polypeptides. 6.1 Structure of nucleic acids and replication of DNA. There are two types of nucleic acid, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. RNA itself has three forms: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. DNA carries the genetic code. Its structure allows it to store information, pass information onto RNA so that proteins can be made, and to copy itself, allowing the genetic code to pass to new cells. Messenger RNA copies the genes to allow them to be used as templates for protein synthesis. Transfer RNA brings amino acids to the ribosome during synthesis. Ribosomal RNA is a major structural component of the ribosome. DNA contains the bases A, C, T, and G. RNA contains the bases A, C, U, and G. A always pairs with T, or U, and C always pairs with G, like A equals T and. 6.1 to 1 Structure of nucleotides including phosphorylated nucleotide, ADP, DNA and RNA are composed of nucleotides, which themselves contain a sugar, a phosphate group and a nitrogen-containing base. DNA and RNA are polynucleotides, made up of long chains of nucleotides. A nucleotide contains a pentose sugar, a phosphate group and a nitrogen-containing base. In RNA, the sugar is ribose. In DNA, it is deoxyribose. Nucleic acids are long chains of smaller units called nucleotides. Each nucleotide has 5-carbon sugar, a phosphate group and an organic base. Deoxyribonucleic acid, DNA, is made of 2 chains of nucleotides. All its nucleotides carry the 5-carbon sugar deoxyribose. Ribonucleic acid, RNA, is made from a single chain of nucleotides. All its nucleotides carry the 5-carbon ribose. 6.1 to 2 Explanation of bases adenine and guanine as purines, cytosine, thymine and uracil as pyrimidines, single-ringed. Adenine and guanine are purines with a double-ring structure and thymine, cytosine and uracil are pyrimidines with a single-ring structure. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. There are 2 groups of organic bases, purines and pyrimidines. Adenine and guanine are purine bases whereas cytosine, thymine and uracil are pyrimidine bases. Adenine, cytosine, guanine and thymine are found in DNA. Adenine, cytosine, guanine and uracil are found in RNA. 3 of the bases in RNA, adenine, guanine and cytosine, are the same as those in DNA. The 4th is different. RNA contains uracil instead of thymine. A DNA molecule consists of 2 polynucleotide chains, linked by hydrogen bonds between the bases. The overall structure is a double helix. In DNA there are 4 bases: adenine, A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. Provided they have the complementary base sequence, strands of nucleic acid can be held together by hydrogen bonds between bases. The base pairs are highly specific. In DNA, adenine always pairs with thymine and cytosine always pairs with guanine. RNA forms similar base pairs, except that adenine pairs with uracil. RNA has only 1 polynucleotide chain, although this may be twisted back on itself, as in tRNA. In RNA, the base thymine is replaced by uracil. 6.1 to 3 difference between RNA and DNA. DNA RNA 1. Double polynucleotide chain. Single polynucleotide chain. 2. Larger molecular mass, 100,000, 150 million, AMU. Smaller molecular mass, 20,000 to 2 million, AMU, 3. Always a double helix. May have a single or double helix. 4. Pentose sugar is deoxyribose. Pentose sugar is ribose. 5. Organic bases are A, T, C, G. Organic bases are A, U, C, G. 6. Ratio of A and T to C and G is 1. Ratio of A and U to C and G varies. 7. Found almost entirely in the nucleus. Manufactured in the nucleus but found throughout the cell. 8. Amount is constant for all cells of a species, except gametes and spores. Amount varies from cell to cell and within the cell, according to metabolic activity. 9. Chemically very stable. Chemically less stable. 10. Permanent. May be temporary and exist for short periods only. 11. There is only one basic form, but with an almost infinite variety within that form. There are 3 basic forms: messenger, transfer and ribosomal RNA. mRNA, tRNA and rRNA, 12. Number of strands, 2 anti-parallel strands. Number of strands, 1. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 6.1 to 4 Semi-conservative replication of DNA during the S phase of cell cycle, the roles of DNA enzymes, leading strands and lagging strands, 5 foot to 3 direction. DNA molecules replicate during the S phase of interphase by semi-conservative replication. Each of the 2 new molecules formed contains one parent strand and one new strand. DNA is copied so that it can be passed from cell to cell during cell division. During semi-conservative replication, the 2 strands in the parent DNA break away from each other. By base pairing, free nucleotides assemble along the exposed single DNA strands. Because the base pairing is specific, each new strand is an exact copy of the old partner strand. DNA polymerase joins together the free nucleotides. Hydrogen bonds between base pairs hold together the 2 complementary strands in a DNA molecule. They also hold parts of RNA molecules together where the single strand has folded back on itself. 6.1 to 5 Ribonucleic acid, RNA. RNA molecules are much smaller than DNA molecules. DNA can consist of over 300 million nucleotides. RNA usually consists of a few hundred. RNA is also less stable. DNA molecules are the permanent store for genetic information and last for many years. In contrast, RNA molecules have a short-term function and are easily replaced. There are 3 forms of ribonucleic acid, RNA, in the cell: mRNA, tRNA, and rRNA. Transfer RNA is found in the cytoplasm and is a carrier molecule, bringing amino acids to the ribosomes for assembly into a new amino acid chain, according to the order specified on the mRNA code. Ribosomal RNA makes up part of the ribosome, a small organelle that brings together all the chemicals associated with protein synthesis. Protein synthesis takes place in 2 stages: transcription and translation. During transcription, a complementary RNA copy of the base sequence of a gene is made. The RNA copy is called messenger RNA, mRNA. In mRNA, each set of 3 bases coding for an amino acid is called a codon. Nucleic acids carry the genetic code that cells use to make proteins. The code determines the sequence of amino acids in a protein. 6.2 Protein synthesis. During translation, the base sequence of mRNA is read by ribosomes and used to assemble a sequence of amino acids. Transfer RNA, tRNA, molecules are involved in this process. They are an anticodon, complementary to each codon that enables them to carry amino acids to the ribosome. The length of DNA that carries a code for one protein is called a gene. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Because many proteins are enzymes, the DNA determines the metabolic reactions that occur in cells. In this way, DNA contributes to the phenotype of organisms. Transcription produces mRNA controlled by the enzyme DNA-dependent RNA polymerase, which unzips the DNA alpha helix. Translation requires two nucleic acid molecules, tRNA molecules to carry amino acids, and rRNA to translate the mRNA strand. Within the ribosome, two tRNA molecules bind to the mRNA, allowing amino acids to form a peptide bond. Immunology and forensic biology. There is a specific and nonspecific immune response. The specific immune response acts against pathogenic microorganisms and viruses. Macrophages in the lymph nodes ingest pathogens and present the antigens on their cell surface membranes. Clonal expansion leads to large numbers of identical B lymphocytes which can destroy a particular pathogen. Plasma cells very quickly produce specific antibodies which bind to the antigen on the surface of invading pathogens. Memory B cells are responsible for a rapid secondary response on further infection. The ABO blood groups, A, B, AB and O, can be distinguished by the antigens on the red blood cells, antigen A, antigen B, both, or neither, and antibodies in the plasma. Everyone has a unique DNA fingerprint. Restriction enzymes cut DNA into fragments. Electrophoresis can be used to sort DNA fragments according to size. Radioactive DNA probes are used to locate specific DNA fragments. The polymerase chain reaction, PCR, can be used to greatly increase the amount of DNA in a small sample. Crop plant cereals are an important food source with high nutritive value. Different kinds of cereals are adapted to grow in different environments. Rice is one of the most important tropical food crops. Rice is adapted to growing partly submerged in water by having air spaces in the stem, enabling the root cells to obtain oxygen for respiration. Flooding favors rice growing because it increases the supply of nutrients and inhibits the growth of competing weeds. Maize is adapted to grow in high temperatures and light intensity. Maize photosynthesizes using the C4 pathway. This enables it to photosynthesize when the CO2 concentration is less than optimal for the C3 pathway, i.e. when it is hot and dry. Sorghum is adapted to grow hot, dry climates that have long periods without rain. The leaves have a thick waxy cuticle to prevent loss of water by evaporation, and roll up in dry conditions, trapping moisture and further preventing water loss. Sorghum is very efficient at extracting water from the soil. The growth of greenhouse crops can be improved by increasing carbon dioxide concentration, temperature and light intensity to certain maximum levels. Carbon dioxide concentration can be increased by pumping in gas or using paraffin bearers. Artificial lighting and heating can increase light intensity and temperature. The most important macronutrients are nitrogen, phosphorus, and potassium. Fertilizers supply nutrients to the soil and improve crop yields. Fertilizers can be inorganic or organic. Weeds reduce crop yields by competing for nutrients. Pests reduce crop yields, for example by damaging plant leaves. Pesticides, fungicides and herbicides can be used to control insects, fungi and weeds respectively. Biological control uses predators or parasites of pests to control their numbers. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Agrochemicals, fertilizers and pesticides damage the environment. Minerals leached from soil contaminate fresh water, leading to eutrophication. Persistent pesticides move through the food chain, becoming increasingly concentrated and possibly reaching lethal concentrations. Manipulating reproduction. All female mammals have an estrous cycle with a follicular stage, ovulation, and a luteal stage. During the follicular stage, the follicle-stimulating hormone, FSH, stimulates a follicle to become a mature ovarian follicle. A surge in luteinizing hormone, LH, brings about ovulation. The same surge in LH causes the follicle remains to become corpus luteum. The corpus luteum secretes estrogen and progesterone. Estrus refers to the changes occurring around the time of evolution. For many female mammals, estrus marks the time when they will allow mating. Human females can control their reproductive cycle using substances that mimic or block the action of hormones. The combined contraceptive pill is an example of negative feedback. It contains estrogen and progesterone which inhibit FSH so that follicles do not mature. The mini pill contains progesterone. Increased knowledge of the human reproductive cycle has led to better diagnosis of and new treatments for infertility. Farmers maximize their animal product yields by controlling animal reproduction. Synchronized breeding of farm animals increases farming efficiency. 6.2 to 1 polypeptide coded for by a gene, a sequence of nucleotides that forms a part of DNA molecule, the main process of protein synthesis. A DNA-based sequence copes on to a mRNA-based sequence during transcription. This sequence is converted into an amino acid sequence during translation. Structure function name of process DNA carries code for all the proteins that a cell can make. None. Messenger RNA, mRNA, copies the code for a single protein from DNA. Carries the code to ribosomes in the cell's cytoplasm. Transcription. None. Ribosome reads mRNA code and assembles amino acids in their correct sequence to make a functional protein. Translation. Transfer RNA brings individual amino acids from the cytoplasm to the ribosome. None. 6.2 to 2 Principles of the universal genetic codes in which different triplets of DNA bases either code for specific amino acids or correspond to start and stop codons. The sequence of nucleotide bases in a DNA molecule codes for the sequence of amino acids in a polypeptide. Each amino acid is coded for by 3 bases. A length of DNA coding for one polypeptide is a gene. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Protein synthesis can be considered in 2 stages, transcription and translation. During transcription, the base sequence of part of a DNA molecule is copied by a molecule of messenger RNA, mRNA. Each group of 3 bases on the mRNA molecule, called a codon, codes for a particular amino acid. This is the genetic code. Specific start codes ensure that the genetic code is non-overlapping. This means that it can only be read in a unique sequence of 3 bases at a time. 6.2 to 3 The importance of DNA in transcription, translation to construct polypeptides including the roles of RNA polymerase, mRNA, codons, tRNA, anticodons, and ribosomes. During translation the mRNA moves to a ribosome in the cytoplasm. Transfer RNA, tRNA, molecules bring the appropriate amino acids to the ribosome. The ribosome moves along the mRNA molecule 1 codon at a time. The sequence of codons determines the sequence of amino acids joined together to form a polypeptide. tRNA molecules bind to the mRNA strand via complementary bases, called anticodons, to those on the mRNA, called codons. The anticodon corresponds to a specific amino acid brought in by the tRNA. Ribosomes are composed of 50% protein and 50% rRNA, and are made of two subunits which remain separate until mRNA is present. 6.2-4 Transcribed, template, strands and non-transcribed strands. Messenger RNA, mRNA, can be thought of as a mobile copy of a gene. Small lengths of mRNA are assembled in the nucleus using a single gene within the DNA as a template. When a complete copy of the gene has been produced, the mRNA moves out of the nucleus to the ribosome, where the protein is synthesized according to the code taken from the DNA. Transcription involves the production of a template of genetic code, and translation is the conversion of the template into a polypeptide. Protein synthesis is the production of polypeptide chains based on the genetic code in DNA. It occurs in two stages in all cells containing nuclei. Transcription is the production of template of the relevant genetic code, and translation is the use of this template to produce a polypeptide chain. 6.2-5 Formation of RNA molecule in eukaryotes during transcription, modified by the removal of non-coding sequence, introns, and the joining together the coding sequences, exons, to form mRNA. In eukaryotes, the mRNA molecule made during transcription, the primary transcript, is modified by the removal of non-coding sequences, introns, and the joining together of remaining coding sequences, exons, to form the final mRNA molecule used in translation. Introns are lengths of nucleic acid that carry a nonsense code. Although copied from DNA to mRNA during transcription, these introns are removed from the mRNA before it is used to make proteins. 6.2-6 Gene mutation, a change in the sequence of base pairs in a DNA molecule that may result in an altered polypeptide. A change in the nucleotide sequence of DNA is a gene mutation. Various types of gene mutation can occur, including base substitution, insertion, and deletion. Spontaneous changes, called mutations, sometimes occur in DNA. These mutations often disrupt metabolic pathways. Cystic fibrosis and phenylketonuria are two examples of disorders caused by defective genes. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 6.2-7 Gene mutation is a result of substitution, deletion, or insertion of nucleotides, results in altered polypeptide production. Key words: adenosine triphosphate, ATP, amino acids, anticodon, antiparallel, complementary base pairing, deletion mutation, deoxyribonucleic acid, DNA, DNA ligase, DNA nucleotide, DNA polymerase, exons, gene mutation, insertion mutation, interphase, introns, lagging strand, leading strand, messenger RNA, mRNA, non-transcribed strand, nucleotide, phosphodiester bond, polypeptides, purine, pyrimidine, ribonucleic acid, RNA, ribosomes, RNA nucleotide, RNA. Polymerase, sickle cell anemia, semi-conservative replication, substitution mutation, transcribed strand, transfer RNA, tRNA, translation, triplet code, universal. Chapter Glossary. Adenosine triphosphate, ATP, is a nucleotide derivative that acts as the energy currency of cells. It consists of a molecule of ribose, a nitrogenous adenine base, and 3 phosphate groups. Amino acids are the monomers containing an amino group, NH2, a carboxyl group, COOH, and a variable R group that make up proteins. Anticodon is a sequence of 3 nucleotide bases at one end of a tRNA molecule that is specific to an mRNA codon. Antiparallel describes the complementary strands of a DNA double helix which run parallel but in opposite directions, 5 foot to 3, and 3 to 5. Central dogma of biology. The process by which the information in genes flows into proteins, DNA, RNA, protein complementary base pairing describes how hydrogen bonds form between complementary purine and pyrimidine bases. 2 bonds form between A and T in DNA or U in RNA. 3 bonds form between G and C. Codon, a sequence of 3 nucleotides that corresponds with a specific amino acid or start/stop signal during translation. Deletion mutation is a form of gene mutation in which one or more nucleotide bases are removed from a DNA sequence. Deoxyribonucleic acid, DNA, is an information-storing molecule made up of deoxyribonucleotide monomers joined by phosphodiester bonds to form a double helix. DNA ligase, an enzyme that catalyses the formation of phosphodiester bonds between Okazaki fragments on the lagging strand during DNA replication. DNA nucleotide is the monomer that makes up DNA and consists of deoxyribose, a nitrogenous base and a phosphate group. DNA polymerase is an enzyme that catalyses the formation of phosphodiester bonds between nucleotides during the synthesis of a new DNA strand. Exons are regions of DNA or RNA that code for amino acid sequences. Gene is a sequence of nucleotides on a chromosome that codes for the production of one or more polypeptide chains and functional RNA. Gene mutation is a change to at least one nucleotide base in DNA or the arrangement of bases. Gene mutations can occur spontaneously during DNA replication. Insertion mutation, a form of gene mutation in which one or more nucleotide bases are added to a DNA sequence. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 03004610-457. Interphase is the longest stage of the eukaryotic cell cycle. It consists of G1 phase, S phase and G2 phase and occurs before mitosis. DNA replication occurs during S phase. Introns is non-coding sequences of DNA found between exons. Lagging strand is the DNA strand that is synthesized discontinuously in the 3 foot to 5 direction, away from the replication fork, during semi-conservative replication. Leading strand is the DNA strand that is synthesized continuously in the 5 foot to 3 direction, towards the replication fork, during semi-conservative replication. Messenger RNA, mRNA, is a type of RNA that carries genetic information from the nucleus to the ribosomes for protein synthesis. It is a single helix consisting of thousands of mononucleotides. Mutation, a change in a genetic sequence non-transcribed strand, is the strand of DNA whose base sequence corresponds to that of the mRNA transcript synthesized during transcription, although it has T instead of U. Nucleotide is the monomer from which nucleic acids are made that consists of a pentose sugar, nitrogenous base and phosphate group. Phosphodiester bond is a type of bond that joins nucleotides together to create polynucleotides. Polypeptides are molecules formed by the condensation of many amino acids. Purine is a class of nitrogenous bases which are made up of 2 rings. Adenine and guanine are members. Pyrimidine is a class of nitrogenous bases which are made up of a single ring. Cytosine, thymine and uracil are members. Ribonucleic acid, RNA, is a relatively short molecule made up of ribonucleotide monomers joined by phosphodiester bonds. Ribosomes are organelles found either free in the cytoplasm or bound to the ER that are involved in the synthesis of proteins. RNA nucleotide is the monomer that makes up RNA and consists of ribose, a nitrogenous base and a phosphate group. RNA polymerase is an enzyme that catalyses the formation of phosphodiester bonds between nucleotides during the synthesis of a new RNA strand. Semi-conservative replication is the replication of DNA to produce 2 new DNA molecules which both contain one new strand and one strand from the original DNA molecule. Sickle cell anemia is a disease characterized by hook-shaped red blood cells. A missense point mutation of GAG to GTG in the HbA, normal, allele for the beta-globin polypeptide results in the HbS, sickle cell, allele of the gene. Substitution mutation is a form of gene mutation in which one nucleotide base is exchanged for another. This may change an amino acid or produce the same amino acid, due to the degeneracy of the genetic code. Transcribed strand is the DNA strand that acts as a template for RNA polymerase during transcription. Also known as the template strand. Transcription is the formation of pre-mRNA in eukaryotes and mRNA in prokaryotes from a section of the template strand of DNA. It is the first stage of protein synthesis. Transfer RNA, tRNA, is a form of RNA that carries specific amino acids to the ribosomes. It is single-stranded and takes a cloverleaf shape. One side is longer than the other. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 03004610-457. Enabling the attachment of an amino acid. At the opposite end is an anticodon specific to the amino acid. Translation is the second phase of protein synthesis that takes place on the ribosomes. mRNA is used as a template for the attachment of tRNA molecules with complementary anticodons. The amino acids carried on adjacent tRNA molecules are joined to form a polypeptide chain. Triplet code is a specific sequence of 3 nucleotides on a molecule of DNA or RNA codes for a particular amino acid in protein synthesis. Universal is a feature of the genetic code, the same codons code for the same amino acids in almost all organisms. Short MCQs with right answers. 1. Which structures contain cytoplasm with mitochondria and a nucleus? Companion cell, option A. 2. What is the correct term to describe intermolecular hydrogen bonding between water molecules? Diffusion. 3. The diagram shows the outline of a xerophytic leaf that had been left for 45 minutes in different conditions, P and Q. 4. Which statements about the cells in layer Y of the leaf in each of the conditions P and Q after 45 minutes are correct? 1. There is a less negative water potential in P than in 2 quetzales. The cells may be turgid in P and plasmolyzed in 3 quetzales. There is no net diffusion of water into Y in either P or Q. 5. How does sucrose move from chloroplasts to the phloem? 1. Diffusion, 2. Apoplast pathway, 3. Symplast pathway. 6. How are companion cells involved in loading sucrose into phloem sieve tube elements? Actively through cotransporter proteins and passively through plasmodesmata. 7. Which row identifies cells with plasmodesmata? Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 03004610-457. 8. Which statements about the apoplast and symplast pathways are correct? 1. In the apoplast pathway, water molecules move through free spaces in the cellulose cell walls of plant roots. 2. In the symplast pathway, water molecules diffuse through the cytoplasm and plasmodesmata of cells. 3. Water molecules traveling through plant tissue move by mass flow along the apoplast pathway. 9. Where does water evaporate from during transpiration? Mesophyll cell walls. 10. The diagram shows the outline of 3 xerophytic leaves of the same type in 3 different conditions, P, R, and S. 11. Which description of the water potential of the cells in layer Y is correct? 12. The photomicrograph shows a transverse section of the leaf of a species of grass. 13. The grass is specially adapted to grow in a dry habitat. Which row correctly explains how the features help the grass to grow in this habitat? 14. Which statement supports the theory of active loading of sucrose into companion cells? 15. Which row correctly explains how the structures of phloem sieve tube elements and xylem vessel elements are related to their functions as regarded as essential for normal metabolism. In sand rats, the gene coding for the PDX1 protein has a much lower proportion of A and T nucleotides than PDX1 genes from other animals. Finding the complete DNA sequence of the PDX1 gene in sand rats has been difficult. Sequencing involves splitting off nucleotides one at a time from single-stranded DNA. What could account for the difficulty in finding the DNA sequence of the PDX1 gene in sand rats? 1. The strength of the hydrogen bonding between the two strands of the PDX1 gene is unusually high. 50. Which statement describes the structure of ATP? It is an RNA nucleotide with two extra phosphates. 51. Which of the following in the key describes uracil? It is not a purine and has a single ring structure. 52. Which statements about thymine nucleotide are correct? 1. Thymine is a pyrimidine. 2. Base pairing occurs with two hydrogen bonds. 53. Molecule of tRNA has the anticodon sequence UAC. What will be the corresponding nucleotide sequence in the DNA? TAC 54. Which row represents the correct features guanine? It is a purine and joins its complementary bases via three hydrogen bonds. 55. Which statements about cytosine are correct? 1. The carbohydrate could be ribose. 2. The organic base contains nitrogen. 3. Base pair occurs with guanine. 56. Which statements about guanine are correct? 1. The carbohydrate can be ribose or deoxyribose. 2. Base pairing occurs with three hydrogen bonds. 3. Guanine is a purine. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 57. Which statements about the nucleotide containing uracil are correct? 1. Uracil is a pyrimidine. 2. The carbohydrate is always ribose. 58. What is correct for thymine? 1. Thymine has single ring structure, is a pyrimidine and joins its complementary base with two hydrogen bonds. 59. Which nucleic acids bases are purines? Guanine and adenine. 60. What is the common component of the three molecules RNA, DNA, and ATP? Phosphate. 61. Which statement about nucleotide is correct? ATP is a phosphorylated nucleotide. 62. Which statement about nitrogenous bases is correct? Uracil is a pyrimidine with a single ring structure. 63. Which statements about DNA/RNA are correct? 1. The DNA nucleotide containing adenine is different from the RNA nucleotide containing adenine. 2. Purines only form base pairs with pyrimidines. 64. Which statements about complementary base pairing are correct? 1. Cytosine forms 3 hydrogen bonds with guanine. 2. Purines and pyrimidines are different sizes. 3. It allows transcription to occur. 65. The statements describe features of some nucleic acids. Which functions are carried out by transfer RNA? 1. Carry an amino acid to a ribosome. 2. Hold amino acids in position for translation. 66. Fill in the blanks with suitable words. DNA contains the pyrimidine base thymine, which is joined to adenine with 2 hydrogen bonds. 67. Which nucleic acid bases are pyrimidines? Cytosine, thymine, and uracil. 68. Which statements about complementary base pairing are correct? 1. Occurs during translation. 2. The base pairs are of equal length. 69. Which statements about complementary base pairing are correct? 1. Purines and pyrimidines are different sizes. 2. It occurs during translation. 3. Uracil forms 2 hydrogen bonds with adenine. 70. Which statements about complementary base pairing are correct? 1. Purines and pyrimidines are different sizes. 2. Adenine forms the same number of hydrogen bonds with thymine and uracil. 3. The base pairs are of equal length. 71. One gene provides the code for the production of which nucleotide? Polypeptide 72. Which gene for protease is activated, which nucleic acid will be formed? mRNA 73. What are the products when a DNA molecule replicates? Two molecules of DNA each made of a sequence of paired nucleotides. 74. Which description of a eukaryotic gene is correct? A sequence of nucleotides that forms part of a DNA molecule and codes for a polypeptide. 75. DNA replication involves several stages. Which statements about DNA replication are correct? 1. Each strand of DNA double helix acts as a template for the opposite strand. 2. Hydrogen bonds between bases A and T and between C and G are broken. 76. What is the role of DNA polymerase? Formation of bonds to complete the sugar phosphate backbone. 77. Which statement correctly describes the base pairing in a molecule of DNA? The purine adenine forms bonds with the pyrimidine thymine. 78. The length of double-stranded DNA contains 120 nucleotides and codes for a section of a polypeptide. What is the maximum length of this section of a polypeptide? 20 amino acid 79. DNA polymerase catalyses condensation reactions between molecules during semi-conservative replication of DNA. Which two molecules are joined by DNA polymerase? Nucleotide and nucleotide 80. What is the minimum number of hydrogen bonds in a length of DNA containing 700 base pairs? 1481. What is the maximum number of hydrogen bonds in a length of DNA containing 700 nucleotides? 1050. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 82. What is the smallest unit of a DNA molecule that can be altered by a mutation and cause a change to the coding of a polypeptide? Nucleotide 83. Which type of sugar and which type of bonds are formed in a DNA molecule? Type of sugar, reducing, type of bond, hydrogen 84. A double-stranded DNA molecule was analyzed and 29% of its nucleotide bases were found to be adenine. What percentage of its nucleotide bases will be cytosine? 21% 85. 21.2% of the bases in a molecule of DNA are cytosine. What percentage would be adenine? 28.8% 86. Which row shows two pairs of nucleotide bases in a molecule of DNA? First base pair, second base pair, bases, present number of H bonds, bases, present number of H bonds at 2CG3. 87. Two polypeptide strands make up a DNA molecule. What is a correct description? The percentage of the cytosine is the same as that of guanine in the whole molecule. 88. Which structural feature of the DNA molecule varies? Number of hydrogen bonds between base pairs. 89. Which type of sugar and type of bonds are found in a DNA molecule? Type of sugar, reducing, type of bonds, covalent and hydrogen 90. What is the maximum number of hydrogen bonds in a length of DNA containing 700 base pairs? 2191. Which statement explains why DNA replication is described as semi-conservative? The template of each new molecule of DNA is one strand of the original molecule. 92. Which features of DNA enable it to meet the requirements as a molecule of inheritance? Requirement of DNA molecule: ability to remain stable, ability to contain information, ability to transfer information, ability to replicate. Sequence of nucleotides: sugar phosphate backbone complementary base pairing, formation of mRNA for translation. 93. which statements about transfer RNA structure are correct? 1. There is a binding site for the attachment of a specific amino acid and a different binding site for the attachment to the ribosome, so that translation can occur. 2. There is a ribose phosphate backbone with strong phosphodiester bonds and areas within the polynucleotide chain where base pairing occurs. 94. Which statements about tRNA are correct? 1. Hydrogen bonds between bases temporarily hold tRNA against mRNA. 2. tRNA translates the base sequence in mRNA into the amino acid sequence in a protein. 95. Which components could be found in nucleotides of rRNA? Any one of two different nitrogenous bases with a double ring structure. 2. Any one of two different purine bases. 96. XNA is a laboratory-made nucleic acid made of nucleotides in which one component has been replaced by chemical X. The chemical X is not found in nature. The part of the molecule responsible for coding is not changed. Which organic component of a DNA or rRNA nucleotide has been replaced by X? 5-carbon sugar 97. Which statements about tRNA are correct? 1. Hydrogen bonds between bases temporarily hold tRNA against mRNA. 2. The specificity of the tRNA molecule for glycan and the specificity of the enzyme that loads glycan are both necessary for correct loading. 98. Electron micrographs may show large numbers of ribosomes forming chains along mRNA molecules. What is the advantage of this arrangement compared to when ribosomes appear singly on the mRNA? Polypeptides can be produced more rapidly. 99. Which statements about tRNA are correct? 1. Contains base pairing. 2. Contains hydrogen bonds. 3. Is single-stranded. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 100. A student produced a table of differences between RNA and DNA. The table contains one mistake. Which row has the mistake? RNA, found throughout cell, DNA, found only in the nucleus 101. Which row shows two pairs of nucleotides formed when mRNA is translated? First base pair translated, second base pair translated, bases present, number of H bonds, bases present, number of H bonds, $2 GC3. 102. A gene codes for the sequence of amino acids in a single polypeptide. Hemoglobin consists of 2 alpha globins and 2 beta globins. How many genes are needed to code for a single hemoglobin molecule? 103. A piece of double-stranded DNA containing 12 by 103 nucleotides is transcribed and translated to produce a single polypeptide. What are the maximum numbers of amino acids in this polypeptide? 2 by 103 104. What is the maximum number of codon-anticodon interactions within one ribosome? 105. In a genetic engineering experiment, a piece of double-stranded DNA containing 6,000 nucleotides coding for a specific polypeptide is transcribed and translated. What is the total number of amino acids in the polypeptides? 1,106. Following translation, the alpha polypeptide hemoglobin, alpha globin, undergoes modification. During this modification, the first amino acid is removed, leaving 141 amino acid residues. How many nucleotides does the gene coding for alpha globin contains? 420. 107. A section of a DNA molecule has the coding sequence AGGCAATGGC. Which statements are correct? 1. Coding sequence contains 3 pyrimidines. 2. The double-stranded DNA of this section contains 26 hydrogen bonds between complementary bases. 108. Which is the correct DNA triplet on the original DNA template that codes for the amino acid histidine, is TCA. 109. The table shows 3 anticodons for different amino acids. Which DNA triplet on the DNA template strand codes for the amino acid serine? TCA 110. A peptide consists of 10 amino acids of 4 different kinds. What is the theoretical minimum number of different tRNA molecules required to translate the mRNA for this peptide? 111. Which statements describe the process of translation order-wise? 1. mRNA binds to the ribosome. 2. tRNA enters the ribosome carrying a specific amino acid. 3. Hydrogen bond forms between the anticodon and the codon. 4. A peptide bond forms between adjacent amino acids. 112. Some antibiotics carrying prokaryotes by binding to RNA polymerase. What effect will this have on protein synthesis? Condensation reactions joining RNA nucleotides will not take place to form mRNA. Amino acid anticodon ala CGU his GUS ser UCA. Amino acid anticodon alanine CGU histidine GUA serine UCA. Lecture notes for AS level biology by Muhammad Rashid Chatta. Cell number 030046100457. 113. What occurs during each of DNA replication and transcription and translation? 1. ATP provides energy. 2. Condensation reactions occur to form a polymer. 3. Hydrogen bond forms between purine and pyrimidine bases. 114. Which statement is correct? During translation, ribosomes join amino acids in a sequence determined by mRNA with the complementary sequence of nucleotides to that of the DNA strand on which it was made. 115. Rifampicin is an antibiotic used to treat tuberculosis. It works by inhibiting RNA polymerase in bacteria. Which of these processes will be directly inhibited by this antibiotic? Transcription 116. The statements describe the features of some nucleic acids. Which functions are carried out only by mRNA? 1. Carry a genetic code sequence out of the nucleus. 2. Carry a genetic code sequence to a ribosome. 117. Ribosome exists as separate subunits that bind together during protein synthesis. What do these subunits consist of? RNA and protein. 118. Which row correctly identifies the features of DNA and RNA molecules? 1. DNA and RNA contain both purine and pyrimidine bases. 2. DNA and RNA both contain a pentose sugar. 3. Hydrogen bonds form between bases in some RNA. 119. Which molecules are involved in transcription and which molecules are involved in translation? Transcription, DNA and mRNA, translation, mRNA and amino acids. 120. What terminates the formation of a polypeptide chain during protein synthesis in cells? When a stop codon is reached on the mRNA molecule. 121. What is the correct sequence for the processes involved in the formation of an enzyme in a cell? Transcription, translation, condensation, ionic bonding. 122. The following statements describe events that take place during DNA replication and transcription. Which row is not correct? The original DNA molecule is changed after the process and no DNA replication and transcription occurs. 123. Which type of molecule is always the end product of transcription? mRNA. 124. What does the process of translation require? mRNA, ribosomes and tRNA. 125. What does the process of transcription require? ATP, DNA and free nucleotide bases. 126. The length of double-stranded DNA contains 120 nucleotides and codes for polypeptide X. What is the maximum length of polypeptide X? 60 amino acids. 127. Which statement describes a process that occurs during protein synthesis? Translation is the linking together of amino acids coded for by mRNA. 128. When a gene mutation occurs, which of the following may be altered, resulting in the production of a non-functional protein? 1. Amino acid sequence. 2. DNA nucleotide sequence. 3. mRNA nucleotide sequence 129. Which statement describes how a gene mutation can lead to the production of a non-functional protein? A codon in the mRNA transcribed from the mutated gene is changed. 130. What is the minimum number of base substitutions required to change the nucleotide sequence of the HbA, normal, allele to the HbS, sickle cell, allele? 1. 131. Which molecule has its synthesis directly controlled by DNA? Amylase. 132. What makes the exact copying of DNA molecules possible? Base pairing 133. A gene codes for the production of a protein, p53, that binds to damaged DNA during interphase and prevents its replication. A carcinogen in cigarette smoke mutates this gene. Which statement explains why this mutation may cause cancer? Lack of p53 allows cells with damaged DNA to replicate 134. The following events occur during transcription before the mRNA leaves the nucleus. Which events will have occurred twice? 1. Bonds break between complementary bases. 2. Bonds form between complementary bases. 135. Which row shows the correct combination? Triplet code, DNA, codon, mRNA, anticodon, tRNA. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 136. Which statement describes migration of DNA? Parental DNA is split into two strands, each of which is replicated. 137. DNA was extracted from a fruit fly and a human cheek cell. In which way did the DNA molecule differ? In the sequence of the nucleotide. 138. The table shows the sugars and some bases found in RNA and DNA. Which is correct? RNA, ribose, DNA, thymine 139. Which statements are correct about DNA transcription and translation? Transcription produces mRNA, translation occurs at the surface of ribosomes 140. In a DNA molecule, the bases sequence AGT codes for the amino acid serine. What is the base sequence of the anticodon on the tRNA to which serine becomes attached? AGU 141. Which diagram shows the bond linking the individual units of a nucleic acid? C-O-P-O, C142. Which statement correctly describes the transcription of DNA? It produces mRNA. 143. In transcription, what is transcribed and what is the product? Transcribed, DNA. Product, mRNA 144. How many different polypeptides, each consisting of our amino acids, can be made if the number of different amino acids available is n? N are 145. A protein contains all the common amino acids. What would be the hypothetical minimum number of types of tRNA molecules needed for the synthesis of this protein? 2146. Which statement about the strands of a newly replicated DNA molecule is correct? One strand is new and the other is part of the original molecule. 147. What is the theoretic number of chemically different dipeptides that may be assembled from 12 different types of amino acids? 144-148. Which enzyme rejoins sections of DNA in genetic engineering? DNA ligase 149. Which structural feature of the DNA molecule varies? The order of bases on a single nucleotide chain. 150. Which type of molecule is the end product of translation? Polypeptide 151. What is the function of enzyme DNA polymerase? To synthesize a strand of DNA. Using DNA as a template. 152. RNA is extracted from beta cells in the pancreas. It is used to make DNA coding for human insulin. Which enzyme is used to make the DNA? Reverse transcriptase 153. A polypeptide consists of 10 amino acids of 4 different kinds. What is the theoretical minimum number of tRNA molecules required to translate the mRNA for this peptide? 454. What is the sequence of bases in mRNA transcribed from this sequence? Ogwakagu 155. The sequence of bases on part of a molecule of DNA is shown. Taka Gaka, sense strand, ATGTTTACTGGT, antisense strand, 156. The following event occurs in the replication of DNA. Arrange them orderwise. 1. Bonds between complementary bases break. 2. Opposite strands separate. 3. Free nucleotides pair with complementary nucleotides on each strand. 4. Bonds between complementary bases form. 5. Sugar phosphate bonds form. Lecture